Well, good morning. Thank you all for joining us here. And we've had wonderful introductions to process validation, and there has been lots of buildup into this surrogate selection portion. So I hope I can answer some of your questions and provide some guidance into that area. I know every year when I'm making my Christmas wish list, I always put a magic crystal ball on there. I'd really like one that gave me the ability to look at something that I'm buying at the grocery store and having a nice little salmonella set up and wave out. Unfortunately, Santa has not brought that to me yet. If you have one, apparently I have not been nice enough. I'll continue working on that. I would also settle for this, a magic ball that could just tell me when something has entered in and potentially contaminated the product. Again, I haven't been nice enough to get one of these quite so yet. But we do have something that we can use that can give us some type of indication whether or not there has been the introduction of an ecologically similar organism to a pathogen. And those are indicator organisms. Indicator organisms are useful for determining if a food has been exposed to a condition that would also increase the risk of contamination by a human pathogenic. Usually we're looking at bacteria in this case, but there is some good recent work that has been done with noroviruses and calciviruses. There was an excellent presentation on Sunday that talked about indicator organisms and the status for that within the water systems and their adaptation to food systems. So if you didn't have a chance to go to that, I would really encourage you to go back to the web and to look at some of those web presentations as well. And for these indicator organisms, there are lots of different ways for looking at that. There are several processes which are direct measures of an organism, and we're all microbiologists and we're used to doing plate counts. Whether or not you choose to do a log transformation of those counts or report your values as colony forming units, whatnot. There's also a number of methods that are using PCR and other ways for either a quantitative or just a yes-no about presence of these indicator organisms. Or from a sanitation aspect, many people are very familiar with indirect measurements such as detection of metabolic end products, especially lactic acids, those types of things for spoilage organisms, and ATP. There's standards that exist for confirming whether or not your process is clean enough or your surfaces are clean enough as you go forward. These indicator organisms can also be really useful for indicating whether or not there was inadequate processing for safety or whether contamination happened after the kill step was put into place as well. So there's really two types of indicator organisms. There's an index microorganism and a surrogate microorganism. The index microorganisms are typically naturally occurring, and that is going to provide an indication that the food has been exposed to something where there's an ecologically similar pathogen. So E. coli is one of the standards for this from an indicator organism standpoint. However, I should point out that their presence alone doesn't indicate that there is a dangerous pathogen within that product. It just means that maybe there was a process failure or there was some type of exposure event that would allow a pathogen that had a similar physiology or a similar type of lifestyle to survive that type of process. So an example you might be familiar with is from a pasteurization standpoint of dairy products. People typically look for the presence of coliforms, yeasts, and molds to indicate that there was a post-process contamination event or also potentially a process failure that led to an inadequate kill within those. And those would be index microorganisms because they're naturally occurring. What has become very popular lately is the use of surrogate microorganisms. So there's a lot of, oops, I'm going forward here. There's a lot of different definitions for surrogate microorganisms, but probably the one that I have found that I like the most is that surrogate microorganisms are harmless microbes with correlated survival and growth parameters to the specific pathogen of interest. And while what is in red was not in the original definition, I'd like to add this into when subjected to the target process conditions, because this is really important. Not all microorganisms respond to all conditions identically. That's earth-shattering news, isn't it? No, we've known this for a long time now. So I'd like to make sure that we all leave this room understanding the importance of knowing that these surrogate organisms behave similarly to the target pathogen that you're trying to inactivate. So we really should be asking the questions about the effectiveness of our control points, also in terms of what are those control points and are the surrogates as responsive to those control points as the pathogen of interest, and specifically within your target food as well. So what do I mean by this? And this is a made-up example. This is not actual validation data. I made it up to illustrate the point associated with this. This is, I can't figure out how to use the cursor, so I apologize. I'm going to lose you on the microphone, I think. So what we're talking here is if you look at the red squares, these are our potential surrogate that is being considered for validation compared to Salmonella. And as you go across the X, this is our temperature increasing, and across the Y, this is our log D value, so increase in time that it takes to have 1 log reduction of either the surrogate or the Salmonella. If you look at the green circle, this shows that it takes more time to inactivate the surrogate in comparison to the Salmonella. So this is great from a processing aspect because you can be more confident that you've inactivated the target pathogen. However, I'd like you to look at starting at the temperature of 175. So that's this red circle here. At this point, it becomes more difficult to inactivate the Salmonella in comparison to the surrogate. And so therefore, at these higher temperatures, the surrogate that is examined here is no longer an appropriate choice for validation work for the Salmonella in case. As I said, this isn't real data, but this does happen in real life because in several cases, Salmonella has more resistance, especially at low water activity processing. So this is definitely something that we need to consider as we go forward. We need to make sure that the process validation is done for the actual parameters that as the process that is being validated. Okay. What are other desirable characteristics for surrogate bacteria? Well, everybody has already alluded to the fact that we want them to be non-pathogenic. We're really trying to keep out pathogens from these food processing plants. So first and foremost, non-pathogenic, harmless. Also, they shouldn't cause spoilage or hygiene issues as well. Number 2, that the inactivation characteristics and the kinetics of that inactivation are comparable to the target pathogen. I really can't stress this enough. The surrogates and the target pathogen should also have similar environmental tolerances. The surrogate should be able to be stable, and it should have good, stable growth characteristics where each time you can reliably achieve the same inoculum and you can achieve the same physiological state, because all of those things will influence the inactivation of that organism. So that leads to having very standardized protocols in place. They should be easily prepared to yield high density populations. That is controversial, as Dr. Schaffner just alluded to there, but typically as microbiologists, if you want to achieve a 5-log reduction, you're trying to inoculate to 7 logs at least in order to have some safety with your limit of detection for that. So usually when you look at these type of surrogate protocol developments, they're looking for creating that 7-log starting density on the population. And also that your inoculum on your product should remain stable until you use it. If you have a 2-log die-off within the time that it's sitting on the shelf and you don't know that, then that's going to seem like, wow, your process is really effective. In reality, it may just be that they weren't very stable on them. So, it needs to know that. And also, you need to take into account that food naturally contains lots of other bacteria there. So you need to be able to discriminate the surrogate from the other native microbiota within that food product of interest. And that can be challenging for certain types of foods. Especially when you're considering in some of the agricultural commodities that have very high microbial counts normally associated with them, like spices. Okay, so there are a number of surrogate microorganisms that have been developed for different target pathogens. In the interest of time, I'm not going to be able to talk about all of them, but I did want to point out that they do exist for Clostridium botulinum. The use of Bacillus amoxicillans and the non-proteolytic Clostridium sporogens is really well established for especially low acid foods, and recently it's been expanded for chilled foods as well. For Listeria monocytogenes, Listeria inoculum is a very popular strategy, and that's been validated for the flask pasteurizations of frankfurters. Also, E. coli K12 is a popular type of surrogate for monocytogenes. That's specifically with non-thermal processing, so looking at E-beam irradiation in cantaloupe that has been performed. For E. coli O157H7, there's a few more choices of surrogates that have been used out there. Again, lots of them that are present for thermal processes and validated for different thermal products, especially ground beef. The meat industry was very active in that role early on, but also most recently for some non-thermal processes like pulsed lights and field treatments of juices and high pressure processing of beef as well. Recently, a lot of the work with surrogates and validation as to whether or not it's an appropriate choice has been with Salmonella. And a lot of this work recently has been with low moisture foods specifically. Some of the organisms that are being used are Geobacillus stereothermophilus. That was some of the early studies looking at extrusion of animal and poultry feed. That is a very conservative surrogate for inactivation of Salmonella. It is very difficult to kill that Geobacillus stereothermophilus. Those of you might be familiar with that. That's a standard surrogate organism used for autoclaving in the pharmaceutical industry for medical implants type of thing, so it's very conservative. Enterococcus faecium, NRRL, B2354, that has had a lot of work associated with pasteurization of liquids, also pasteurization of almonds. That is the surrogate of choice of the Almond Board of California. And it recently has also been validated for extrusions of balanced carbohydrate protein meals. Pantowea agglomeraans has been used also for dry roasted almonds. I would like to point out that that is a potential concern, as it can cause some diseases in individuals who are immunocompromised, so that should probably be used with caution, and is also potentially a plant pathogen as well. For Pediococcus acidolicticae, there's been a lot of work with the Pediococcus groups, specifically within the meat industries, for both beef and turkey jerkies, and also recently with dried pet food as well. And Staphylococcus carnosus, that has been recently validated as a conservative surrogate in ground beef and Frankfurt batter. So I want to again re-emphasize, oh no, I'm already 5 minutes in left. Okay, so I'm going to have to go a lot faster here. So I'd like to re-emphasize again this thermal, the importance of verifying that the thermal resistance, in this case for this ground beef example, is comparable between the target pathogen and the surrogates that are being used. So this is data from Ma et al. In 2007, and it shows the inactivation, so the D value of Listeria monocytogenes and Salmonella sneftenberg, oops, I'm missing a 775W in comparison to Enterococcus faecium and Pediococcus parvalis. And what you see is it takes a lot longer to inactivate the Enterococcus faeciums and the Pediococcus in comparison to the two targets. And depending on who you talk to, they're either worried about that or they're much more confident about that. So what this means is that you're going to be over-processing that product. It's more processing than you need to kill the target pathogen, but it also introduces a better level of safety associated with that product. So it's, again, the level of risk management that needs to be incorporated into there. There's also a very big importance on making sure that the surrogate is tested for the product and for the formulation of that specific product that is being made. So this figure demonstrates that between ground beef and Frankfurter batter, that the survival of E. coli 0157 and non-0157H7 tax is different depending on the composition of that food product. And the predictability of the surrogates that are being tested is different as well. So that's the importance of knowing that what your composition is of your product and assuring that your inactivation are comparable. Also, you need to make sure that you're comparing your surrogates and your target pathogen at relevant processing conditions. So this is a figure that is coming out of Zhang et al. 2011 that's comparing Enterococcus 2354 versus Salmonella enteritidis. And I'd like you to see that the kinetic behavior between the two organisms is very different at 90% moisture versus 5% moisture. So you need to make sure that they're behaving under the conditions that are relevant to the process at hand. Also, we need to consider the tolerance of the pathogen and the inoculation strategy for those as well. This is data that's coming out of my lab for my project that is funded by ILSI North America, where we're looking at the stability of different surrogates on black peppercorns, and we compared 3 different inoculation preparation methods. We compared the stability of Salmonella and Enterococcus faecium B2354, and we find that that inoculation preparation does influence the stability of both the surrogate and the target pathogen. So you have to be very careful in how that inoculation is prepared and how it is applied. Okay. Also, we find too that inoculation preparation influences the subsequent inactivation. And that's not only for the target pathogen, it also is applicable for the Enterococcus as well. And probably most importantly, how we prepare that influences whether or not it's considered a good surrogate. So this is data for whole peppercorns that are exposed to saturated steam processing for 92 seconds. And this first set, this biofilm inclusion here, means that the organisms are allowed to become part of a native biofilm where they're adherent to other cells. Black peppercorn has about 8 logs of other bacteria on there, so we don't know who exactly they're interacting with. We just know that there's lots of them there. And when you treat them to saturated steam, again, trying to achieve that 5-log reduction, and for this case, we can achieve that biofilm inclusion here, but we achieve greater reduction of the Enterococcus faecium using that method. In comparison, when they're grown on plates, on tryptic soy agar plates, and scraped from those plates according to the methods proposed by the Almond Board of California, the Enterococcus faecium is a good surrogate for that because you have more reduction of the salmonella in comparison to the Enterococcus faecium. So it's really important, and maybe we also need to start considering creating the worst case scenario in our inoculum preparation method as well, or at least having it as close to the type of contamination events that would be occurring for your product of interest. For low moisture foods, dry carriers have had a lot of interest recently. This is work that was recently published by Inoculum et al. In 2015, where they examined the effectiveness of dry carriers and again how that inoculation preparation influenced the inactivation, in this case the D value with time. They compared Salmonella tennessee versus the Enterococcus faecium. And while the Salmonella tennessee, under their conditions, there wasn't a difference based on inoculation preparation and the inactivation, there was a difference for the Enterococcus faecium, B2354. So maybe under these situations, it's better to use a liquid grown before applying them to the dry talc, and then applying that and incorporating that within a matrix here as well. But I would like to point out with the dry carriers, there are some concerns and some problems transferring adequate cell densities from a carrier to a particle of interest. And there's also, if you include that dry carrier into the product itself, that can influence the thermal dynamics within that as well. So you need to consider how appropriate is that for your particular product. It would work great for some types of products, but not as well for others. Other considerations? Again, I think we've all talked about this, we all understand this. We want to make sure that any surrogates, anything that is introduced into a plant for validation work should not impose a health risk. I know several years ago there was a lot of concern about the Enterococcus faecium in our RRL B2354. It had been reclassified as a BSL-2 organism because of the fact that, oh, aren't all Enterococcus faecium exactly the same? No, in this case, this particular strain that has been used has been verified to not contain the virulence genes that are necessary for human infections. That was published in Kopit et al. In 2013, and subsequently it was redesignated as a BSL-1 status. Also consider whether or not these surrogates are going to create any problems associated with loss of quality for the product. So I know that can be of concern, especially with the lactic acid bacteria that are used. Lactic acid bacteria make a much better choice for things like low water activity foods because you don't have high enough water activity for them to start wreaking havoc in the system. But for meats and other systems, that is something that needs to be considered before those would be used. Also, you need to understand how your plant hygiene and sanitation system works. Are they going to automatically start having lots of positives for some type of their hygienic indicators because that surrogate has been there? And also, potentially from a regulatory implication. There had been a lot of interest in trying to use attenuated strains for in plant use because physiologically the organisms are going to be the same. But isn't one salmonella the same as another salmonella in the regulatory world? I'll leave Dr. Zink to address that with you. But that's just a point of consideration. I would like to acknowledge everyone, firstly, the ILSI North America Food Microbiology Committee for funding this spice work and for inviting me to come speak with you today. And I'd also like to acknowledge my team. They have designated themselves Team Spice. I'm so proud. But these are the graduate students and technicians that have been working on this project, and I would be remiss in not acknowledging their work on here. This is Brett, Jordan, Chris, me, Natalie, and Kim. So thank you all for your attention. And I do apologize. I think I went slightly over, but do we have time for questions?
Okay, we have time for at least one question. If anybody would like to go to the center mics.
Katie Swanson, thank you for a comprehensive discussion, but a thought just occurred to me. We've got quite a broad audience here. The title of this symposium is Surrogates for Process Control,
Sure.
But none of the titles of the individual presentations are talking about that. When you made the comment that you should try to shoot for like 10 to the 7th or something for inoculation study, I sat there and went, well, what if you're looking at control of growth? And then you can overwhelm the population. So I just would like your thoughts on that and also clarify for everybody here, this is about killing critters, not preventing them from growing.
Yeah, you're very correct in that aspect. Is the title is not necessarily reflective, but when I went back to it and I thought, okay, this is about how do you inactivate, or how are you validating that your inactivation works, not that something like a preservative is inhibiting growth in the future. Yes, thank you.